Another major hormone that is produced by the pancreas is glucagon. To start, glucagon is a 29 amino acid peptide hormone secreted from the alpha cells of the pancreas. It plays an important role in maintaining the glucose homeostasis. So it is also produced in other sites other than the pancreas. So we have here the structure of glucagon. So glucagon is again a 29 amino acid peptide hormone, and it is because it is a peptide hormone, it has two ends. So the other end here represents the free, the N-terminal amino acid residue, while the other end here represents the free carboxyl group, which is also called as the C-terminal amino acid residue. So we have here another representation of the structure of glucagon. So glucagon is a single polypeptide, and it has no disulfide bridges, making it incredibly flexible. So for the history of glucagon, discovery of glucagon occurred only 2 years after Banting and Bess discovered insulin. The first notion of glucagon came when John Murlin and co-workers detected that pancreas extracts contain a second hormone with the ability to increase blood glucose. They gave it the name glucagon from its function as a glucose agonist. Glucagon, unlike insulin, is considered to be a catabolic hormone. Both insulin and glucagon play a central role in maintaining glucose homeostasis. So glucose homeostasis is represented by a blood glucose level of 90 to 110 milligram per deciliter. So glucagon induces the liver to deliver glucose through glycogenolysis and gluconeogenesis. It is also important in providing glucose to the brain in hypoglycemia. So the glucose is considered to be the only nutrient that the brain and the RBC can utilize as a source of energy. So we have here a diagram showing how the glucagon and insulin will act in order to promote the glucose homeostasis. So during high blood glucose level, the pancreatic beta cells will be stimulated to secrete the hormone insulin, and insulin will act in order to maintain the blood glucose level to normal. During a low blood glucose, the pancreatic alpha cells will be stimulated to secrete the hormone glucagon, and glucagon will act in order to restore the normal blood glucose level. We also have here another diagram showing the mechanism whereby insulin and glucagon maintain the blood glucose level. So the red one here represents the action of insulin, while the blue one here represents the action of glucagon. So for insulin to be secreted, we have here a stimuli that is a high blood sugar level. So this particular stimuli will promote the release of insulin from the pancreatic beta cells. So upon release, insulin will act in order to stimulate the uptake of glucose from the blood towards the tissue cells. Insulin will also act on the liver in order to stimulate the production of glycogen from glucose. So glucose will be formed from glycogen will be formed from glucose. So this action here of insulin will lower the blood sugar. A low blood sugar will serve as a stimuli to the pancreatic alpha cells to secrete the hormone glucagon. So glucagon will act on the liver to stimulate the breakdown of glycogen. So when this will happen, it will raise the blood sugar level. So what are the target tissues of glucagon? So the target tissues of glucagon are those tissues with glucagon receptors. Example of these are the hepatocytes and the adipocytes. The muscles do not have receptor for glucagon. and it does not contribute to increased blood glucose in hypoglycemia. So there are two major sources of glucagon. The first one is, of course, the pancreatic islet alpha cells. You also have the L cells. So the L cells are highly specialized gut endocrine cells. So this include, for example, we have the stomach. The stomach produces the gut glucagon. That is identical to the pancreatic glucagon. Another is the small intestine. It also produces an immunologically similar molecule called the glycentine. So aside from the L cells and the pancreas, the other minor sources of glucagon include the neurons in the brainstem and the hypothalamus, as well as the salivary gland. For the glucagon synthesis, so glucagon is produced as a prohormone with 160 amino acids in the rough endoplasmic reticulum of the alpha islet cells. It is cleaved in the RER into 4 peptides, including glucagon. It is then stored as a secretory vesicle in the cytoplasm of the alpha islet cells. It is then released in response to a major stimulus, which is hypoglycemia. So after that, it is metabolized mainly by the liver and the kidneys, and it has a half-life in the plasma of about 5 minutes. So we have here a diagram showing the synthesis of glucagon from pro-glucagon. So glucagon is derived from precursor pro-glucagon, which can be processed into a number of related peptide hormones. The pro-glucagon contains glucagon, the glucagon-like peptides. We have the GLP-1 and the GLP-2. We also have the GRPP, which stands for the glycentine-related polypeptide, and the intervening peptide 1 and the intervening peptide 2. The pro-glucagon is expressed in different tissues, so it can be expressed in the pancreas, the pancreatic islet alpha cells. It is also expressed in the intestinal endocrine L cells, particularly found in the ileum, large intestine, duodenum, and the jejunum. And to a minor extent, the pro-glucagon can also be expressed in the neurons in the brainstem and in the hypothalamus. The processing of the pro-glucagon is undertaken by the processing enzymes, the prohormone convertase 1,3 and the prohormone convertase 2. So in the pancreas, the PC2 or the prohormone convertase 2 will process the pro-glucagon to glucagon, while processing the pro-glucagon in the intestine in the brain is undertaken by the PC1-3. Again, in the pancreas, the pro-glucagon will be processed into glucagon, glycentine-related pancreatic polypeptide GRPP, intervening peptide 1, and the major pro-glucagon fragment or the MPGF. In the intestine and in the brain, the pro-glucagon will be processed by the PC1-3 into glucagon-like peptide 1 or the GLP-1, glucagon-like peptide 2 or the GLP-2, and the oxyntomodulin and the glycentine, as well as the intervening peptide 2. So we have here a diagram showing the tissue-specific processing of pro-glucagon in the pancreas and in the intestine. So in the pancreatic alpha cell, the pro-glucagon is processed by the prohormone convertase 2 activity into GRPP, glucagon, and the major pro-glucagon fragment or the MPGF. In the L cells of the intestine, the pro-glucagon will be processed by the PC1-3 into GLP-1, GLP-2, and the glycentine. By further PC1-3 activity, the glycentine can be subdivided or further cleaved into the glycentine-related polypeptide, the GRPP, and the oxyntomodulin, as well as the glucagon. So we have here a diagram showing the structural organization of our pro-glucagon, as well as the pro-glucagon-derived peptides. So when we say pro-glucagon-derived peptides, this includes our glycentine, our oxyntomodulin, and our MPGF. So peptides that are liberated specifically in the pancreas or in the intestine are indicated below the full length for the pro-glucagon sequence, along with their amino acid sequence and the therapeutic potential. So the numerals above the pro-glucagon structure indicate the relative amino acid positions of the pro-glucagon-derived peptides within the pro-glucagon. So for example, our glycentine here is represented in these amino acid positions of our pro-glucagon. Our oxyntomodulin here contains the glucagon and the intervening peptide 1, and it is located at this amino acid position. Our MPGF, or the major pro-glucagon derived fragment, is located in this amino acid position, and it contains these peptides here, the GLP-1 or the glucagon-like peptide 1, intervening peptide 2, and the glucagon-like peptide 2. So the processing of our pro-glucagon in the pancreatic alpha cells will produce our MPGF, or the major pro-glucagon derived fragment, as well as our glucagon. While the processing of our pro-glucagon in the intestine, particularly in the L cells of the intestine, will produce the peptides glycentine, oxyntomodulin, GLP-1, and GLP-2. So take note that the glucagon is contained within our oxyntomodulin and our glycentine. So the glycentine and the oxyntomodulin contains the peptide hormone glucagon. So GLP-1, so this is the amino acid sequence of our GLP-1. So the H here represents histidine, and the G here, for example, represents the amino acid glycine. So what is the therapeutic potential of this GLP-1? So GLP-1, or the glucagon-like peptide 1, is now used as a major drug class for the treatment of type 2 diabetes mellitus. So this GLP-1 drug here, which is the Rebelsus, this is the first glucagon-like peptide receptor protein treatment that is approved for use in the US for the non-insulin treatment for people with type 2 diabetes mellitus. The GLP-2 here is used in the clinical trials for the treatment of Crohn's disease and the short bowel syndrome. So the Crohn's disease is a type of inflammatory bowel disease. And this is the amino acid sequence of the GLP-2. For glucagon, this is the amino acid sequence of glucagon, and the glucagon receptor antagonism target is used for the treatment of type 2 diabetes. So what are the factors that will stimulate glucagon secretion? So the major factor that will stimulate the glucagon secretion is hypoglycemia, particularly blood glucose level that is less than or equal to 70 milligram per deciliter. It also includes fasting or starvation and exercise. Another factor that will stimulate the glucagon secretion is the protein-rich diet. Amino acid is considered to be a stimulatory regulator of our glucagon secretion. So another is the GIP hormones, particularly the gastrin and the CCK or the cholecystokinin, as well as the GIP or the glucose-dependent insulinotropic peptide. Another factor that will stimulate the glucagon secretion is the parasympathetic nervous system. What are the inhibitors of glucagon secretion? So this includes hyperglycemia, particularly a blood glucose level that is more than or equal to 160 milligram per deciliter. Another factor is the hormone insulin and the diet that is carbohydrate rich and fat rich, including glucose, ketone, and fatty acids. So we have here a diagram showing the glucagon secretion, particularly the glucose-dependent glucagon secretion from the alpha cell of the pancreas. So the major stimulus for this particular mechanism is hypoglycemia or low blood sugar level. So during hypoglycemia, the intracellular glucose concentration will fall with a subsequent production in the glycolysis generated ATP in the mitochondria of the cell. So because there is a reduction in the ATP level within the cell, this will result to the closure of the ATP-sensitive potassium channels. The closure of the ATP-sensitive potassium channels will cause the accumulation of our cation here, our potassium, within the cell. Accumulation of potassium in the cell, because potassium is a cation, it will cause the depolarization of the cell membrane. Depolarization of the cell membrane will result to the formation of the action potential, and this will trigger the opening of the calcium channels, particularly the voltage-dependent calcium ion channels. Opening of the voltage-dependent calcium ion channels will result to the influx of calcium ions, and this will increase the intracellular calcium concentration within the cell. The calcium here in the cell will trigger the secretion of glucagon through the process of exocytosis. So this diagram here represents the glucagon concentration in response to hypoglycemia, euglycemia, and hyperglycemia. So in this diagram, this diagram here represents the plasma glucose level during hypoglycemia, euglycemia, and hyperglycemia. The x-axis here represents the time, while the y-axis represents the plasma glucose in millimoles per liter. So in hypoglycemia, the blood glucose is at about 2 to 5 millimoles per liter. So during euglycemia, it is about 5 millimoles per liter, and during hyperglycemia, it is about 12 to 13 millimoles per liter. So this diagram here on the other side represents the plasma glucagon concentration during hypoglycemia, euglycemia, and hyperglycemia. So during hyperglycemia, you can notice here that the glucagon concentration is below 10 picomoles per liter. So during the exercise or during hypoglycemia, you can notice here that the circulating glucagon levels increases dramatically. So there is a 3 to 4 times increase in the basal levels of glucagon secretion, increasing the glucagon to insulin ratio. So as you can notice here, now this one here represents the basal level of the glucagon secretion, while this one here represents the glucagon secretion during fasting or during hypoglycemia. So as you can notice here, the glucagon secretion reaches its peak to about 40 picomoles per liter, which is about four times the basal level, which is about 10 picomoles per liter.